So hello students, welcome to Aakash. I'm your botany faculty Aman Vishwakarma, and today we are here to discuss the AIATS plus phase 2 of mock test 6 paper that was conducted on 23rd of Feb 2025. And from the previous session you will know that here we are going to discuss those questions where most of the students have made mistakes. Okay, and for the session I'm going to discuss questions that belongs to these topics. Sexual reproduction, the topics that are present here are sexual reproduction, organism and population, plant kingdom, morphology, okay, cell the unit of life, and photosynthesis. So on the screen the first question is question number 127. The spread of living pteridophyte is limited and restricted to narrow geographical regions because of, okay, so to understand the pteridophytes limitation with respect to the geographical regions, we need to understand the life cycle of pteridophytes. Okay, so let's start and study some life cycle of pteridophytes. Okay, so basically pteridophytes are also called as the first terrestrial plants containing vascular tissues. Okay, so these are the first terrestrial plants with vascular tissues. Okay, now as you have seen the previous life cycle of bryophytes, the main plant body was gametophytic, but in case of pteridophytes you will find that the main plant body is diploid and sporophytic. Okay, so here in case of pteridophytes you will find that the main plant body is a sporophyte. Now this sporophyte is diploid. Okay, now the sporophyte body consists of true root, stem and leaf structures. Okay, and the sporophyte is also carrying sporophylls. Okay, now this sporophylls, consist of spores. These spores, the sporophylls are present either on the leaflets or the leaves. Okay, now you will see sometimes in pteridophytes there are some leaf-like structures on the abaxial region. You will find that there is presence of some dot-like structures. These are called as sorus, or they contain spores. Okay, so these are the sporophylous structures that you see in the pteridophytes. Okay, now these sporophylls will now produce spores. The ploidy of the sporophyll is also diploid. Now the sporophyll, which consists of spore mother cells, will develop, divide into spores by the process of meiosis. So there is a process occurring which is called as sporic meiosis. So here the spores ploidy will be haploid. Okay, now most of the pteridophytes are homosporous. That means the spores that are formed are either of same morphology or same type, or it can be different, right, like heterosporous. Okay, condition. So most of the spores that are produced in case of pteridophytes are homosporous, but some examples of heterosporous pteridophytes are Selagina and Salvinia. Okay, so now the spores will develop into prothallus. This prothallus is also a gametophytic structure. Now prothallus is inconspicuous, inconspicuous but small and multicellular structure, which is also green and photosynthetic. This gametophyte is having a specific requirement, and the specific requirement is that it requires cool, damp and shady place. Okay, so the specific requirement of gametophytic stage of pteridophyte is it requires cool, damp and shady regions. Okay, apart from this specific requirement, they also require water for process of fertilization. Okay, so these two requirements that you see here for gametophyte is the main reason why the pteridophytes are restricted to specific regions or localities. Okay, now you will find that the prothallus will develop. The prothallus or the gametophytic structure may contain female and male sex or male gametophytic structures, right? So if a particular species of pteridophytes is homosporous, then the male and female gametophyte will be present on the same plant. That is, the condition is monoecious. Okay? While as if we talk about heterosporous species, the gametophyte will either, if the spores are formed from male spore or from different heterosporous condition, then there is a gametophytic structure where one contains male gametophyte and the other plant or other gametophyte contain female gametophytes. Okay, so here you will find that there is a dioecious condition. Okay, so here it can consist of antheridia and archegonia. Okay, the male and female sex organ will contain antheridia and archegonia. So here antheridium male and archegonia female. Okay, now the antheridium will form anthrazoids or male gametes which are flagellated. Now for the process of fertilization or for the process of syngamy, okay, for the process of syngamy, they require a medium which is water, and this specific requirement is what is hindering the pteridophytes to not spread into wider localities. Okay, so here after syngamy they will form zygote, and after zygote the embryo will form and leads to sporophytic phase. That was all about the life cycle of pteridophyte structure. So let's see what's the answer of the question. Okay, question number 127. Okay, so the spread of living pteridophyte is limited and restricted to narrow geographical region because of dependence of gametophyte on sporophyte. No, because gametophyte is a green photosynthetic structure, so it is a free living structure, it is not dependent on the sporophyte. So first option is incorrect. Next option says gametophyte that require cool, damp and shady places to grow. Yes, it's the main reason why pteridophytes are not spread throughout the terrestrial land or terrestrial localities. Okay, so this option is true. Third option says no need of water for fertilization. No, because here the male gametophytes will produce male gametes that require water for the process of fertilization. Okay. Fourth option says the sporophyte which can grow only in dry condition. No, pteridophytes are majorly found in cold, damp, shady places, but some pteridophytes are also there which are also present in sandy soils. Okay, so this is not the correct option. So here for this question the correct option will be option number 2. Okay, so let's move to our next question. Yes, so the next question belongs to question number 110, which belongs to sexual reproduction in flowering plants, and there are certain structures that are given in column 1 and the column 2 comprise of their features. Okay, so we need to match the structure with their specific features. So let's study the embryogeny or the embryo structure of monocots as well as dicots so that we can answer this question which is on the screen. Okay, so you will find that in case of after process of fertilization, the embryo will develop into the zygote will develop into embryo. Okay, now for the process for the embryo development, there is certain amount of endosperm needed for the nourishment of the embryo. Okay, so in plants or in angiosperms, you will find that endosperm development precedes the development of embryo. Okay, now if we talk about dicots, if you talk about dicots, you will find that the structure comprise of two cotyledons and an embryonal axis. Okay, now you will find that these two cotyledons in the dicot's embryo structure, and the tip region that you see here, is a plumule. and below the region you will find epicotyle, and I have to extend this part, just a second. Okay, so you will find that below the epicotyle you will find hypocotyle. Now the hypocotyle will terminate into radicle, and the radicle can be covered by root caps. Okay, so let's see the main diagram that we see in the NCERT. Okay, so on the left hand side you are seeing a monocot embryo, while as on the right hand side you are seeing a dicot embryo. Okay, now let's try to identify each structure one by one. Okay, so tell me, the structure that you see here is the structure that you see here is a cotyledon. Okay, in case of monocots it's cotyledon. Now it is, monocots are of single cotyledon, and the single cotyledon is called as scutellum. While as if you see the dicot, so this is a monocot and this is a dicot. Now if you see the cotyledons, these are the two cotyledons that you see in case of dicots. Okay, so you will find in case of monocots you will find that these two structures are coleoptile, and the coleoptile covers the plumule region of the embryo. Okay, while as the lower portion that you see here is coleorhiza, which is covering the radicle. Okay, and radicle is covered also by the root caps. Okay, while as in monocots you will see some shoot axis as well. Okay, so and sometimes in case of grasses you will find that there is another structure which is called as epiblast. Epiblast is the remnants of the second cotyledon that was lost due to the duration of the evolution. Okay, so it is a epiblast. While as if you see the dicot structure you will find that this portion is the plumule. Below the plumule you will find a cylindrical structure which is called as hypocotyle. While as below the hypocotyle you will find that it terminates into a radicle, and the radicle is covered by root cap. Okay, so this was the basic detail and structure of the embryo in case of monocots and dicots. When you talk about the embryogeny of dicots and monocots, there is certain similarity if we talk about the process of embryogeny. Okay, so in the process the zygote will develop into proembryo. Okay, and then the proembryo will develop into globular embryo, and then heart shape. In case of dicotyledons you will see that the heart shaped structure, heart shaped embryo is formed, and then the mature embryo is formed. Okay, so this particular portion is common for both monocots and dicots, while as the heart shaped structure is for the dicotyledons and furthermore. Okay, so let's see the question. Column 1 says in epiblast is a remains of the second cotyledons. Okay, while as the coleorhiza encloses the radicle and root cap. Hypocotyle is plumule stem tip. Okay, plumule is a stem tip and hypocotyle terminates at its lower end in the radicle. So here for A3 is matching. Okay, so this option is incorrect. For A3 is matching, so this option is also incorrect. Now option number 2 says A for 3, okay, and Coleoriza for B for 2. No, this is also incorrect. So here B is true, B1, A3, C2, okay, C2 and D4. So here our correct answer for this question will become option number 4, okay. Shall I move to the next topic? Yes. Now the another question that is very much asked in the NEET as well is about the factors that affect the process of photosynthesis, okay. And the factors that you see in overall environment, you will find that there are only two kinds of factors that you find. First is the intrinsic factor of a plant, while as the extrinsic factor of a plant, okay. So when I talk about the intrinsic factor, when I talk about the factors that affect our photosynthesis, that affect the process of photosynthesis, so there is two factors that affect the process of photosynthesis. First factor is the intrinsic factor. Of plant, okay. Now the intrinsic factor is dependent on the genetic predisposition of the plant. What's genetic predisposition? That means it depends on the genes that are present in the plant, and it can code for various factors. Like if we talk about certain factors, you will find that it can be responsible for number of chloroplast, okay, aging of leaf or age of leaf, okay, type of or chlorophyll pigment presence or abundance, okay, chlorophyll presence, and one major factor which is the internal carbon dioxide concentration in case of plant. Internal CO2 concentration is one of the important factor which is considered as an intrinsic factor in case of plant. Now another factor that you see in case of factors that affect the process of photosynthesis is the extrinsic factor. Now basically the extrinsic factor comprise of light, temperature, carbon dioxide and water, okay. So it comprise of light, temperature, CO2, atmospheric carbon dioxide and water, okay. So we will talk about the external factors that are responsible for the rate or for hindering the rate of photosynthesis, okay. In the next slide you will see that you have known the law which is called as Blackman's law, yes. So for what Blackman's law says that if a particular process is carried out by more than one factor, then the factor which is limiting will be the responsible one which will hinder the process itself, okay. So if I talk, let's take an example. There is a plant, okay, and in the plant the light is limiting, okay. So there is not enough light, while as the other factors, so this particular graph is for light intensity and there is a rate of photosynthesis, okay. So in this condition all the other factors, that is CO2 concentration, temperature, okay, these are constant. The rate of photosynthesis is now limiting for the light intensity, okay. So as the intensity of light increases, the rate of photosynthesis increases, while as at certain point you will find that now there is a limitation or saturation at that point, and now the graph will show a linear graph, okay. That means this point that you see here on the graph is the saturation zone, saturation point. Now light is not the limiting factor here. Now what maybe what's the possibility that now carbon dioxide in the atmosphere is a limiting factor. Okay, so after the saturation, as the plant is not able to increase its rate of photosynthesis even after increasing the light intensity in the environment, now the plant may have been lacking in some other factors like carbon dioxide or the temperature. Okay, so it can be carbon dioxide or it can be temperature. So that's what the limiting or the Blackman's law says. Okay, so for the light intensity, you will find that as there is a low light intensity in the environment, the rate of photosynthesis will increase as we increase the concentration of light intensity itself. Okay, but after a certain point, you will find that the light intensity have no effect in the process in the rate of photosynthesis. Okay, so this is called as saturation point, and in this point you will see that there are other factors that become limiting. Okay, so basically in nature you will find that light is not a limiting factor. Okay, because the 10% of sunlight is enough for the process of photosynthesis. But there are certain cases where you will find that some plants like herbaceous and shrubs plant where the canopies of large trees are shading the sunlight. Due to the shading, the light is very much rare for them, so the light become limiting factors. Okay, so if we talk about just a second. Now if we talk about light, so you will find that herbs, shrubs growing in a forest will have light as a limiting factor. Now light itself can be increased, but high amount of light intensity will damage the pigments of the chloroplast, and this will lead to the decrease in the process of photosynthesis. Okay, so that was all about the light itself. Now let's move to the concept of carbon dioxide as a limiting factor. Now in environment you will find that the concentration of carbon dioxide is around 0.03 to 0.04%. Okay, that much amount of carbon dioxide is present in the atmosphere. Okay, which we can also term it as 380 parts per million or 380 microliter per liter. We can also state it like that. Now in the environment, this much amount of carbon dioxide is present. Okay, so there are certain plants which are having different carbon dioxide saturation point. So here you will see that in case of this graph, you will find that as the concentration of carbon dioxide for C3 plants, okay, as the concentration of carbon dioxide in C3 plant is increased, okay, at certain point the saturation will occur, and now the carbon dioxide is saturated. This point is beyond the atmospheric concentration. Okay, this is the atmospheric concentration, which is around 380, in between 380 microliter per liter. Okay, so in case of C3 plant, you will find that the graph exceeds the or the saturation point exceeds the atmospheric carbon dioxide concentration. Okay, so this is the region or the saturation point, and the region is about 450 microliter per liter. Okay, while as if we compare this C3 plant with the C4 plant, you will find that the saturation point of C4 plants reaches before the atmospheric concentration, because the saturation for C4 plant is 360 microliter per liter. Okay, so basically for C4 plants, carbon dioxide is not a limiting factor, while as for C3 plant, carbon dioxide is a limiting factor because atmosphere में उतना concentration नहीं है for C3 plants, like because C3 plants require 450 microliter per liter of carbon dioxide for fulfilling the saturation point, so that it can perform the optimum rate of photosynthesis. Okay, so for C3 plants here, for C3 plants, carbon dioxide is a limiting factor. Okay, we can use this. We can use this property of C3 plant to our advantage, right? You have seen greenhouse, right? In greenhouse, what happens? The carbon dioxide concentration, or there is a closed space, and the carbon dioxide concentration is quite high as compared to the atmosphere. So if you grow the C3 plants like bell pepper and tomato, the productivity of that plant will increase, right? And due to which you will get higher yield. So you can use this property of C3 plant for producing higher biomass. So like bell pepper and tomato, we can use this property of bell pepper and tomato to increase their biomass in the greenhouse. Okay, now that was all about the concept of carbon dioxide as a limiting factor. So you need to understand that carbon dioxide is the major limiting factor because in environment the atmospheric concentration of carbon dioxide is 380 microliter, or around 0.03% to 0.04%. Okay, so due to its atmospheric unavailability, C3 plants is limiting for carbon dioxide, while a C4 plant doesn't. Okay, now the next concept is of temperature. Why temperature is playing as a factor for affecting the rate of photosynthesis? You have learned the biochemistry, and in biochemistry you have learned the enzymes, right? And in case of enzymes, you have find that enzymes work at different temperatures according to their protein properties, right? So similarly, in case of photosynthesis, you have learned different enzymes, right? Like peptidase. Just a second. You have learned that there are different enzymes like peptidase, Rubisco, ATP synthase, synthetase, right? These are all the enzymes that are taking part in the process of photosynthesis. Yes or no? Yes? Okay. So these are all enzymes and they require their optimum temperature. So whenever a condition arises where the temperature, where the light intensity, carbon dioxide is very much abundant, but still the temperature is very low, then the process have no effect, or the process of photosynthesis will have no effect, the rate will not increase. Because temperature is required, or temperature is required for the optimum activity of the enzymes. So for the temperature itself, you will see this kind of graph where the enzymes, they will form a this kind of graph. Okay, in this case you are finding that there is an optimum range. Okay, this is the optimum temperature where the enzyme will act and will carry out the highest rate of process or highest rate of catalysis. Okay, so here this kind of graph can be seen in case of temperature. Now C4 plant are quite adapted to dry tropical regions, and because of this and also of their adaptability of higher temperature, they are quite favorable for higher temperature, while C3 plants are not so. Okay, so this was all about the discussion of the factors that affect the photosynthesis. Why I am not talking about the water? Because water is not directly affecting the process of photosynthesis. Though water is quite important because water can act as a medium, right? Water is very important for our life, but because of water stress condition, you will find that plants' leaves get wilt, and due to wilting of leaf, the surface area decreases, and due to the surface area decrease, the process or the area or the process of photosynthesis will also indirectly decrease. Similarly, due to water stress, stomata will close, and due to closure of stomata, the gases are not going to enter, and so the process of photosynthesis will be hampered. So directly the water is not affecting the photosynthesis process, but it is affecting the plant, due to which the factors is getting affected or rate of photosynthesis is affected. Okay. So let's try and solve the question that is asked from the previous slide. Question number 107. Read the following statements, state them as true or false, and select the correct option. Okay. So try to answer the first option, that is, there is linear relationship between incident light and carbon dioxide fixation rate only at high light intensity. Okay. At high light intensity you will find saturation, right? Even after if you increase the light intensity and carbon dioxide, you will find that there is no effect. Okay, so this option is incorrect. Carbon dioxide is the major limiting factor for photosynthesis. Yes, it's the major limiting factor for photosynthesis, while as the C option says dark reaction in photosynthesis is more temperature sensitive than light reaction. Yes, in this part, in case of light and dark reaction, you will see in dark reaction there are various enzymes that are involved in the process of synthesizing carbohydrates, right? And for the synthesis of carbohydrates, multiple enzymes are involved. So temperature plays a key role for optimizing the process of photosynthesis. Okay, so dark reaction in photosynthesis is more temperature sensitive than light reaction. Okay. Option D says the effect of water on photosynthesis as factor is more through its effect on the plant rather than directly. Yes, this option is also true, because water does not directly affect the rate of photosynthesis. What it does, it affects the plant physiology, and due to the physiological damage to the plant, the process of photosynthesis is also hampered. Okay. So for this question, A is false. A is false. Okay. B is true, so this option is incorrect. A is false, B is true, no. A is false, B is true, okay. C is also true, and D is also true. So our correct answer will be option number 3, while the fourth option is ruled out because A is said here, it's true, okay. So I hope this concept of factors of photosynthesis was clear to you, okay. Shall I move to the next slide or the next question, okay. So let's see the next question. So the next question is again from the photosynthesis, and the concept here that is asked is splitting of water molecule occurs on the inner side of the thylakoid membrane, and NADP reductase enzyme is located on the lumen side of the thylakoid. Okay, so this is basically about the chemiosmotic hypothesis in photosynthesis. Okay, so let's try the understanding of chemiosmotic hypothesis so that we can give the correct answer for these statements. Okay. Now, this is the thylakoid membrane. There is a similarity if you talk about both chemiosmotic hypothesis and in case of oxidative phosphorylation. The similarity is that both processes produce ATP. What's the common point is the main reason or the main point is for synthesizing ATP. ATP synthesis is a common point, okay. But in case of oxidative phosphorylation, the organelle that was involved was mitochondria, okay. So here, as the mitochondria, which is a double membrane bound organelle, okay, I am trying to draw the mitochondria here. This is the outer membrane and this is the inner membrane, okay. Okay. Now, the hydrogen concentration or the accumulation of hydrogen ion was carrying out in between this membrane, that is perimitochondrial space. This location is where the abundance of hydrogen ion is occurring, okay. And this gradient is formed, the proton gradient is formed in the perinuclear space, and now the proton gradient is breaked and a channel which is basically a complex 5 consisting of F0 and F1 particle will now produce the ATP. The same concept is carrying place in the chloroplast. What's the basic difference here is that the accumulation of hydrogen was carrying out in between the perinuclear space of mitochondria. While as if we talk about the chloroplast, you will find that in chloroplast the thylakoid membrane, yes, this is the thylakoid membrane. In the thylakoid membrane, the hydrogen ions are accumulating in the lumen side of the in the lumen side. So here the protons are accumulating in the lumen side. That's the difference about the mitochondrial accumulation in case of oxidative phosphorylation and in case of chemiosmotic hypothesis. Okay, so let's start the process of chemiosmotic hypothesis. Okay, so here there are various photosystems, right? And in the process of non-cyclic photophosphorylation, you have observed that there is presence of oxygen evolving complex, yes, which is associated with photosystem, yes, photosystem 1 or 2, which component is responsible for oxidizing the process, yes. So here it is associated with photosystem 2. So oxygen evolving complex, which is present on the inner side of the thylakoid membrane, will break the water into oxygen and protons. Okay, these protons are going to be accumulated in the lumen region, while as the photosystem, which is going to be excited by the light, will transfer the electron that is obtained by the water splitting and transport it to the plastoquinone. Okay, plastoquinone, which is a hydrogen acceptor, will transport the hydrogen from stroma to the lumen while transporting the electron from photosystem 2 to cytochrome B6. Okay, the plastoquinone will now transport the electron to the cytochrome B6, and the cytochrome B6 will now transport the electron to the plastocyanin, which is present on the inner side of the thylakoid membrane. The plastocyanin, which is a mobile electron carrier, will transport the electron from cytochrome B6F to photosystem 2. Okay, and in the photosystem 2, you will find that the FDR or FNR will transport this electron to the NADP plus, H plus, and convert them into NADPH. Okay, so you will find that the hydrogen that is present in the stroma is also being utilized for the formation of NADPH. So there is different points here. How many points? Let's see. So a hydrogen is removed from stroma by production of NADPH, which is formed by the enzyme called as NADPH synthetase dehydrogenase. Okay, so here which is called as NADPH dehydrogenase. Okay, which is located on the stroma side of the thylakoid membrane. इधर से रिमूव हुआ, ठीक है, हाइड्रोजन इज रिमूव फ्रॉम द स्ट्रोमा from this point, while as the hydrogen is now again removed due to the transport of electron from photosystem 2 to other photosystem. So there is also removal of hydrogen due to the movement of electron or the transport of electron due to the movement. Okay. Movement of electron through the thylakoid membrane. That's one point, right? And another hydrogen is accumulating because of splitting of water, right? So due to splitting of water as well, splitting of H2O, water is also accumulating in the lumen. So there are various hydrogen ions that are now accumulated in the lumen. Now after breaking the concentration gradient, that is of hydrogen ion from lumen, what will happen, the lumen will now transport from high concentration to low concentration into the stroma region, and this transport is by the membrane, integral membrane protein, which is called as CF0. Okay. while as this is the F1 portion of ATP synthase, which have ATP synthase activity. Okay, so due to the movement of hydrogen ion from the integral membrane protein, there is enough energy generated that causes the conformational change in the F1 part. Okay, and due to this conformational change, the ADP in presence of inorganic phosphate will convert into ATP in the stroma. Okay, so basically all this process, all this thing for accumulating the hydrogen ion in the lumen was for the process of synthesizing the ATP. Okay, so this was all about the story of chemiosmotic hypothesis. Okay, I hope the chemiosmotic hypothesis was clear. Now let's see the question. Splitting of water molecule occurs on the inner side of the thylakoid membrane. Yes, as you can see, the splitting of water is carried out in the lumen side. Okay, so here on the inner side of the thylakoid membrane. So this is true, while as the statement 2 says the NADP reductase enzyme is located on the lumen side of the thylakoid membrane. No, as I have said, the NADP which is formed on the stroma side, okay, is caused due to the NADP reductase, that is enzyme present on the stroma side of the membrane. Okay, so it is not present on the lumen side. This is incorrect. So here second option is also incorrect. Okay, so here only statement 1 is correct, while as only this is also incorrect. Both statements are correct. No, both statements are incorrect. Okay, so here the answer will be option number 1. Is that clear? Okay, now let's move to the next question. So the next question is from Cell, the Unit of Life. In human beings, the membrane of an erythrocyte has approximately 40% lipids and 52% proteins. Yes, this statement is true because as the electron microscope observation, we have found that in case of RBC, the concentration of lipids in human RBC, you will find that the concentration of lipids and proteins are different. The proteins are 52%, while as lipids are 40%. Okay. The plasma membrane discovery was quite intriguing because after the electron microscope discovery, the plasma membrane clear structure was able to be identified. We found that the plasma membrane consists of majorly lipids, consists of lipids and proteins. Okay, so it comprises of lipids and proteins. Now the lipids that you see in case of plasma membrane, you will find that lipids are phospholipids, that they are phosphorylated, and the lipids consist of polar head and non-polar tail. Okay, non-polar tail. Just a second. You will find this kind of lipids, okay, different, that are arranged like this. Okay, so these phospholipids consist of polar head and non-polar tail. This non-polar tail is made up of saturated hydrocarbon Some reasons consist of, if you are seeing here, there is a bending. This bending is because of the pi bond or the double pi bonding. Okay, this is because the pi. Okay, so here the lipids comprise of polar head and non-polar tails, while as the proteins can be classified into integral membrane protein or the peripheral protein. This was discovered because on the basis of the accessibility of the protein or easy processing of protein. So if I am carrying out a process where I am destroying the plasma membrane, okay, and the protein that is removing very easily it was called as peripheral protein. Okay. While as those proteins that are present or were difficult to remove were called as integral protein. Now term, if I try to draw the diagram of plasma membrane, so this is a lipid membrane, okay. There are various lipids, okay. Okay, this is all lipid structure. Now in lipid structure you will find some proteins that are present on the periphery, okay, and some proteins are present inside the membrane. Now this integral membrane protein can be partially embedded in the membrane, like some are partially embedded, or can be fully embedded in the membrane layer, right, lipid bilayer. So that was what the discovery was all about. Now comes the Single and Nicholson, who gave the fluid mosaic model of plasma membrane. They said that lipids are the quasi-fluid nature of lipids provide the fluidity to the membrane. Okay, so their concept was that a plasma membrane comprised of sea of lipids on which the proteins are floating, on which proteins are present. Okay, so that was the concept about the fluid mosaic model, where quasi-fluid nature of lipids is providing the fluidity. Now, due to which the proteins are able to freely move in the lipid sea. Okay, so this was all about the lipid structures and all. Okay, so here the integral and peripheral protein lie on the surface of membrane. So no, this option is incorrect, because as I have discussed in this slide that they are divided into peripheral and integral, and peripheral proteins are present on the periphery, whereas the integral proteins are present either embedded in the plasma membrane or can be partially embedded in the plasma membrane. Okay, so C option says that quasi-fluid nature of lipids enable lateral movement of proteins within the overall bilayer, which is true, right? The fourth option says the fluid nature of the membrane helps in functions like cell growth, cell division and formation of intercellular junctions. Yes, this is the fluid nature of plasma membrane also provides help in functions like cell growth, cell division, formation of intercellular junctions. Okay, so this option is also true. So here the correct answer will be A. A is true, B is false, true. So T, F, T, T. Okay, so T, B is false, so this option is incorrect. A is true, B is false, okay. C is true and D is true, so this option is true. A is true, B is false, but C is not false, okay. So this option is also incorrect, and the fourth option is A4, which is incorrect. So here our correct answer will be option number 2, okay. Let's move to next question. The next question is from Morphology of Flowering Plants. You are seeing different floral structures here, right? And each structure are indicating certain positions of ovary with respect to the thalamus, okay? Which of the given diagram represents epigynous condition in guava and cucumber? Now, there are different types of flower on the basis of the position of ovary on the thalamus and with respect to the other voles, okay? So let's try to understand each and every type of flowers here. So this flower that you see here is hypogynous flower, while as these two are perigynous flower, and this is epigynous flower. Okay. Now, what's the difference between all these flowers is that you will find that the ovary, these ovaries consist of other floral whorls, right? So here the other floral parts, that is androecium, I am writing androecium here, androecium, petals, sepals, right? They are all originating below the ovary. They are all originating from below the ovary, due to which this kind of flower are also called as inferior flower, because the ovary is located at the upper position, while as the other floral parts are present at below the ovary. So, some examples that you see for hypogynous flower is brinjal, China rose, mustard. Okay. Now comes the perigynous flower. In case of perigynous flowers, the ovary and the position of the other floral whorls are looking like they are occurring from the periphery or the side of the ovary. So, here this kind of flowers are perigynous flower, and the examples are peach, plum and roses. Okay. Whereas in case of epigynous flower, you are seeing that the ovary is enclosed, okay, and the other floral parts are arising above the ovary. Okay. So, their position is superior than the ovary. So, this kind of flowers are called as inferior or inferior plants, okay, inferior flower. As the ovary is located at the basal position, while the other floral parts are present above the thalamus. Okay. So, here some examples that you see are guava, okay, cucumber and ray florets of sunflower. Okay. Now, on the basis of this data, you can now solve the question: which of the following diagram represents diagrammatic epigynous condition as in guava and cucumber? Okay. So, epigynous flower is look like this. This is the epigynous flower. So, here it's a perigynous flower, it's epigynous flower, it's also a perigynous flower. So, here it is the correct answer, that is, it is the epigynous flower, while as there are perigynous and it is hypogynous. Okay. So, I hope this question was correct or question was clear to you. Yes. Let's move to the next question, that is question number 94. Okay. So, the 94 question is from: which of the following pairs of organisms store food in the form of compound that is very similar in structure to amylopectin and glycogen? So, this question is from plant kingdom, and we have to answer whether these organisms are containing some storage material which are very similar to amylopectin and glycogen. So, let's try to re-revise the concept here, that there are different classes of algae. Okay. So, names here: first is Chlorophyceae, second is Phaeophyceae, third is Rhodophyceae. Yes, these are the three classes of algae. Yes. Okay. So, these are the different classes of algae. Now, Chlorophyceae is also called as green algae. Okay. So, they are green algae. So, they comprise of chlorophyll A, B. Okay. If we talk about Phaeophyceae, so Phaeophyceae is also called as brown algae. Okay. And in brown algae, you will find chlorophyll A, xanthophyll and C. Okay. While as in case of Rhodophyceae, you will find that they are called as red algae, and in red algae chlorophyll A, D and phaeophytin, which provides the red pigment to the Rhodophyceae members. And phaeophytin, sorry. So, here the storage material for all these classes, you will find that in Chlorophyceae the major storage food is starch, while as in case of Phaeophyceae you will find mannitol and laminarin as a food material. Okay, mannitol and laminarin. While as in case of Rhodophyceae you will find that They store Floridian starch. Now Floridian starch structure is quite similar to amylopectin and glycogen. Okay, so the question was focusing on the structure on the or the storage structure that look like amylopectin and glycogen. Yes, so our answer will be for we are going to see which of the given organisms belong to Rhodophyceae member, because you have learned that in case of Rhodophyceae, Floridian starch is formed, which is structurally similar to the amylopectin and glycogen. Okay, so Polysiphonia and Porphyra belongs to red algae. Okay, yes, this option is true. Ectocarpus and Dictyota belongs to brown algae. This option is incorrect. Cara and Volvox belongs to green algae. So again, it's incorrect. Fucus and Sargassum, again brown algae. So this option is also incorrect. Yes. So here for our question, the correct answer will be option number 1. Was that clear? Yes. So I hope for today's session, it was quite a detailed session. And for the previous session, you will see various topics that you will find intriguing. Okay. For now, prepare your NEET exams and stay hydrated, stay fresh. Okay. Thank you.